William C. Campbell received a BA from Trinity College, University of Dublin, in Ireland, 1952, and a PhD from University of Wisconsin, Madison, Wisconsin, in the United States in 1957. He was with the Merck Institute for Therapeutic Research from 1957, and in 1984, he became the Senior Scientist and Director for Assay Research and Development. Campbell is currently a Research Fellow Emeritus at Drew University, Madison, New Jersey, USA. Please join me in welcoming Dr. Campbell, who will present his Nobel lecture under the title Ivermectina, a reflection on simplicity.
Your Excellencies, ladies and gentlemen, as you see from my title, I intend to talk about simplicity. Simplicity in the context of science. And in talking about simplicity, I do not mean to suggest that science itself is simple, nor will I suggest that the discovery and development of the drug ivermectin was an exercise in simplicity. I want rather to call attention to the element of simplicity within science. And I want to do that by pointing out the prominence of simplicity in the genesis of ivermectin. It has, of course, long been acknowledged and recognized that simplicity has an intrinsic appeal to scientists as it does to others. And indeed, simplicity is widely celebrated in science as a matter of beauty. But I want to talk about simplicity not as a matter of beauty, but as a matter of practical utility. Consider a real-life, actual event. On a particular day, the 9th of May, 1975, there was a mouse in a mouse box in a laboratory. It had been purposely infected with worms, but not enough to cause illness. And on that particular day, the mouse's diet was altered. It was altered by having some liquid stirred into its regular food. And it ate that food for almost a week. And then its regular diet was restored. And almost a week after that, its worms were gone. The mouse had been cured of its worminess. And from that moment, there was a train of events set in motion that would lead to an advance in medicine, both human medicine and veterinary medicine. And that advance in the basic science would in turn lead to practical changes in the management of diseases. So to a very large extent, the drug ivermectin was brought about by simple science. It was not conventional science, it was not obvious science, but it was simple science. I want to make a slight digression here in relation to another question. I have often been asked in recent weeks how it felt when I heard that I had won the prize. And I can say without hesitation that my mind was instantly flooded with two emotions, one an emotion of joy and gratitude, and the other emotion, one of sadness. And the sadness because I knew instantly that all the people who contributed to this drug and made it a success could not be named individually. And so I am a representative of the Merck and Company research team. And in that capacity, I am grateful and honored beyond imagining. So, what was in that liquid that was added to the regular food of that mouse on the 9th of May? It was a liquid in which a microbe, a bacterium, had been allowed to multiply, breaking down the surrounding nutrients and producing all kinds of byproducts, metabolites of various kinds. In other words, the liquid that was added to the diet had been fermented by a bacterium. And that bacterium had been isolated and sent to Merck and Company by Dr. Satoshi Omura, whom you have just heard, and who shares with me the prize that has brought us here together today. And many years later, I had the pleasure of visiting him in Tokyo. Microbes do not all look alike or act alike, but the Kitasato Institute and Merck, both of them, were interested in finding microbes that stand out from the crowd. Microbiologists get tired of finding microorganisms that have already been found. Professor Omura sent us microbes that were unfamiliar in general to microbiologists. And we found that one of those unfamiliar microbes produced an unfamiliar substance, and that that substance had very potent activity against worm parasites, and also against some other parasites, but that is a story for another time. The mouse that I mentioned was a single mouse. I do not mean that the mouse was unmarried. I mean only that that special diet had been tested in only one mouse. Usually things are tested in groups of mice, experimental groups, but that was tested in just one single mouse. Other individual mice got other diets altered by other liquids in which other bacteria had flourished, but only one mouse, only one of those liquids ended in the cure of worms. Just get some water here. Excuse me for a moment. That test method, which might strike you as unusual, was developed in the parasitology department of Merck and Company by Dr. John Egerton. And that is also where, in the same department, Dr. Dan Ostland had pioneered the reduction of experimental group size to a singleton. Simplicity in the history of ivermectin was just a beginning. From then on, there was complexity, many years of basic, complex research, and many years of complex developmental research. Pharmaceutical development is the epitome not of simplicity, but of complexity. And many scientists and many disciplines were involved in the development of this drug. And I will mention perhaps one or two, perhaps biased towards parasitological matters, but I will have, I trust, in the written version of this talk, opportunities to acknowledge my colleagues and family and friends more generously. Many things were going on at once at this time. The microbiologists described the bacterium as a new species of Streptomyces. The fermentation chemists and biologists isolated the mystery substance that killed worms, and they persuaded the microorganism to produce that substance more abundantly. And analytical chemists determined its chemical structure. It's actually a set of closely related molecules that we named ivermectin. The synthetic chemists found how to make an even better version of it, and so I will show you the structure. This is a picture I took of Dr. Omura in 1990, and you can see that he has not changed at all. This is the original stuff, ivermectin, or ivermectin B1A. This is a simplified slide. There's a lot of detail left out of this. It's also known as abamectin. So that's the original stuff, unmodified. But our organic friends in chemistry, they acknowledge that microorganisms can make molecules that humans cannot, but they also seem to believe that only a chemist can make it properly. And so they tweak these things and modify it, and so what they did was to make a better version of it, which is shown here. Now, I trust you all have noticed the difference, but just in case you haven't, just focus on the upper right-hand corner, where you see here and here, the 22-23 bond between those carbon atoms. Each of them has been given another hydrogen. So this was accomplished by hydrogenation of ivermectin to make ivermectin. And so the ivermectin is 22-23 dihydroivermectin B1A. B1A just being one of these closely related molecules. So they had made this change by adding hydrogen. So they thought, not surprisingly, that they would like to call this stuff not ivermectin, but ivermectin, because hydrogen had been added. But it was pointed out to them that in some language, hyver means testicle. And so that's how ivermectin became ivermectin. Despite being unique in its origin, ivermectin now has many relatives. Many of them are on the market in animal health. And there's a whole family of these related compounds called macrocyclic lactones, and there is vast literature on those. Meanwhile, the parasitologists had found out which worms it would kill, and the biochemists found out how it would kill, or rather how it would paralyze them. Because with parasitic worms, paralyzing them is just as good, or from the worm's point of view, just as bad, as killing them. The body itself will get rid of paralyzed worms. With this flood of information that was being gathered, the project was given developmental status, so even more disciplines, more scientists became involved. And I will just mention one as an example of many very different scientific disciplines. And I want to mention for a moment the veterinary parasitologists. We had many veterinary parasitologists, not only from our own country, but from veterinary schools all over the world. And they were brought into this project. And they had an astonishing breadth and depth of knowledge of parasites. And they set up trials to assess the efficacy of ivermectin against many different kinds of parasites in many different kinds of host animals in many different lands. And I think their expertise was actually essential to the success of ivermectin in animal health, where it became the dominant antiparasitic agent. Sometimes things that one are thought of as bad turn out to be good. If a broad-spectrum antiparasitic agent fails to be active against some particular important pathogen, that is usually oblivion for that drug candidate, if it misses some very important parasite. But not necessarily so. In the case of dog heartworm, the the significant pathogen is the adult worm stage, and ivermectin is not active against adult heartworm, Dirofilaria immitis, in dogs. However, in the routine treatment of dogs to get rid of their worms, and I emphasize in the routine treatment that goes on all the time, it is actually dangerous to kill that adult worm. Killing the worm, the adult worm, is quite likely to end in the death of the dogs. It can be fatal to dogs. It can be fatal to the reputation of the veterinarian. And so that is not a good thing to do. So ivermectin turns out to be ineffective exactly where you would like it to be ineffective. Still, ivermectin is used all the time to control heartworm disease in dogs. It's used as a preventative agent. And as it happened before the discovery of ivermectin, I had instituted a program for heartworm research at Merck. And Linda Blair and I had set up the life cycle in the lab, mosquito to dog life cycle, and we had that going just when we needed it to test ivermectin against heartworm. And we found that it's active against a pre-adult phase, so we were able to do the research that led to the development of a once-a-month treatment, the first once-a-month treatment for the prevention of heartworm in dogs. The potential value of ivermectin in humans was never overlooked. I had always insisted that our written objectives include the development of drugs for the control of parasites of people. And when my colleagues, John Egerton and Linda Blair, found that ivermectin was active against the larvae of the parasite Onchocerca cervicalis in the skin of horses, then I knew it was time to take further action. And my chief, Dr. Jerry Birnbaum, enthusiastically approved my proposal to invite Dr. Bruce Copeman in Australia to do a trial against another onchocercal species, Onchocerca gutturosa, in cattle, and we gave him funds to support that research, which he did, and he's an expert on it, and again he found that ivermectin was effective. So at that time, we had information suggesting that ivermectin might be useful in humans against a variety of parasitic worms, including hookworm, for example. But it was clear when one would evaluate the situation that the best way to meet an unmet need in human medicine would be to have it used against river blindness, which is caused by yet another species of Onchocerca, Onchocerca volvulus. So Dr. Birnbaum and I took that message to the top level of research management at Merck and Company. It was an exciting moment for both of us. We made our pitch, and at that time, the head of research at Merck was Dr. Roy Vagelos, who himself was a physician and a chemist. And they listened very intently to our story, and they approved a trial of ivermectin in humans, a very cautious trial in patients that had the very early stages of river blindness, long before there was any actual blindness. They would have the infection that causes river blindness, but they would have light early infections. So this was a very cautious trial at the beginning, and this was one of a series of trials that was set up and directed by Dr. Mohammed Aziz, who was one of Merck's clinical directors, and he was in charge of doing this. And he set up this trial in Senegal, and it was undertaken by Dr. Aziz and Dr. Diallo of the University of Dakar and Dr. Lariviere of the University of Paris and their colleagues. And they showed that ivermectin was active against the microscopic larvae, the baby worms, in the skin of people. And that was, of course, turned out to be exactly what is needed. The trial had to be very cautious because there had been some precedent that it was dangerous to kill those microscopic worms, but this turned out not to be dangerous and, in fact, to be very safe. And to understand why the activity of that against those larvae is important, it's necessary to understand that in river blindness, unlike the situation with dog heartworm, it is the offspring, these microscopic progeny, that cause the damage to the skin and to the eye, and eventually leads to blindness, as well as skin disease and serious itching. And so killing those larvae can prevent the onset of disease. You can block it before it gets going, and that's what ivermectin does. In 1987, Dr. Vagelos was then CEO, president of Merck & Company, and he announced that the company would donate the drug for the use against river blindness. And this led to an unprecedented donation of an unprecedented translation of this donation of the drug into the actual distribution of the drug. And that was a very complicated business, and it was undertaken by many groups. I will just mention a couple of them. You've heard of some of them already. But I mentioned the World Health Organization and Merck's own Mectizan donation program, which was set up from the very beginning to make sure that it was properly used, and the Carter Center in Atlanta, Georgia. But there were many, many other agencies involved in this process. And also there were many leaders involved in this process, many directors of the various components. And I will just mention one of them. And I feel I must mention one because he is Swedish. He is Dr. Björn Tilifors, and I am honored that he is here for the activities of this week. I will now show just a few more slides. This is a sculptural representation of a well-known, iconic scene in endemic areas, where you have endemic areas that are endemic for river blindness, where you have adult people who are blind being led by children and often connected with a stick, sometimes 3 or 4 or 5 people all connected to one child. And this is a well-known thing, and this is where the river blindness program comes in. And the impact of ivermectin and river blindness in terms of the extent of that impact, which has now been developing over many years, is something that is now well documented and well established, and not for me to discuss here. I just want to show a few pictures of some of the very early treatments that I was an observer for in the early program. This is Dr. Mohammed Aziz, the Merck clinical director, who was responsible and who chose the place to do the first trial, chose the place to people to do it with, and made sure that all was done according to proper protocol. I'll show another slide of people. There's 3 people here of great significance. My wife and I are talking to 2 of the people. We're on the left, and I don't need to say why my wife is important. You already know that. But the 2 people on the right are of tremendous importance. One is Jimmy Carter, the head of the Carter Center, former president of the United States. And he, because of his past position, was in a position to be very influential in helping the distribution program. And then standing next to him, second from the left, second from the right, and this picture is Dr. Roy Vagelos. And he was the one who had to make the decision what to do with this drug, and he was the one who decided it would be donated. And there is no point really in talking about various people who have suggested it in various parts of the world at various times, because the important thing is who considered it, who were in a position to do something about it. If you have to be accountable for it, then it becomes a big decision to make, and that's why Roy Vagelos made such an important decision. Now, I would like to not talk further about how great an impact this was, but rather I want to get back to that mouse assay, the test system with which I began. The operation of that assay was simple, as I have explained. The operation was simple, but so was the principle of it. Thinking it up was a different matter. That was innovative thinking on the part of my colleagues. but the underlying principle was simple. It was bizarre, but it was simple. And I have described it this way. You line up a series of infected mice. You treat each mouse with an unknown amount of an unknown substance that might not be there. And then you check to see if it worked. On the face of it, it seems to fly in the face of what we are taught about science with its well-regulated system and its emphasis on measurement. But we need to understand that this, too, is well-regulated science. Empirical research based on observation and trial and error and screening of substances has been the foundation of the discovery of antiparasitic drugs. But in recent decades, that screening approach has been beaten into disrepute and desuetude and has in many places been abandoned. And when it is abandoned, we cannot know what price has been paid in non-discovery. I have recently made a proposal. Before I get to that proposal, let me just show these last few slides. I had a chance to observe the early trials. This is in a remote village in northern Togo, where I saw the first community-based trials in remote places like this, where villagers would gather together in a schoolyard or under the tree for treatment. You see on the left a girl standing on a bathroom scale to be measured, because at this stage very detailed records were kept. It turned out later that that bathroom scale was an unnecessary expenditure of high-tech instrumentation, and it was long since replaced by a measuring stick, which is much easier to use and much more efficient. Children were given a tiny pill, which they could easily swallow and have a drink of water and open their mouths to show that they had swallowed it. This is a village elder. When you, in one of these gathering situations where the treatment was being given, when you would see people like this, a blind elder who had the leopard skin caused over many, many years of infection with river blindness and had been preceded by really excruciating itching. Unfortunately, in those situations, you would also see a man like this, a blind man for whom treatment had come too late, because ivermectin will prevent blindness, it will not cure it. But what the good part of those collections of visit under the tree or in a schoolyard was seeing collections of children who would now be safe and would no longer grow up under the threat of the skin disease and the itching and the eventual blindness. I recently made a proposal for more broadly searching the world for natural products, especially microorganisms, as a means of finding new molecules for development as chemotherapeutic agents. And that might be useful to supplement to some of the mass treatment programs that are currently underway to treat various diseases. But nevertheless, chemotherapeutic control should not be seen as the ultimate objective. Unnatural measures are likely to have unforeseen natural consequences. And the broader the activity spectrum of a biodynamic substance, the more we must guard against the hazards of indiscriminate use. And since we cannot count on discriminate use, we should try to control the disease without recourse to chemical agents. There are very few acceptable anti-parasite vaccines, but we might yet learn how to make better ones and more of them. And we also might be able to devise public health measures to subvert parasite life cycles more subtly than in the past. In late afternoon, I often climb a nearby hill. Not a mountain, it's just a grassy half-mile hill called Half Mile Hill. And from the top, there is a marvelous vista of woodland and lake and the sky often tinted with color as evening falls. And it is a moment of uplifting tranquility. But one realizes, of course, that not everybody lives amidst such natural beauty and in conditions of peace. And around the world, there are many people making heroic efforts to redress that terrible imbalance. And one of their objectives is to improve global health. And as we bring science to bear on such problems, I hope we will keep in mind that solutions are sometimes to be found in science that is simple. Thank you.